My name is Aisosa Omere, and today I will be discussing the utilization of diazo-protecting group strategies in chemical biology to optimize the current proteomic workflow. Protecting group chemistries are an incredibly underutilized tool in chemical biology. Protecting groups are a class of molecule that temporarily mask the characteristic reactivity of a particular functional group that could interfere with the intended reaction. They are easy to put on, remove, and are inert to reaction conditions. They come in a wide variety of forms that can facilitate orthogonal chemistries. This proposal seeks to improve upon the current proteomic workflow by utilizing chemistries that already exist to promote greater efficiency and accessibility in the field of top-down proteomics. Top-down proteomics describes the field of proteomics that studies the intact protein. The advantages of this are that the intact protein retains its structural information, as well as the information gained through post-translational modifications. The class of protecting group that this proposal focuses on is the diazo compound. Diazo compounds are much smaller than their analogous azeto compounds and display a broader range of reactivity. They are relatively easy to make and show amazing chemoselectivity for dipolarophiles such as alkynes, alkenes, carbonyls, and nitriles. In protein modification experiments, diazo compounds selectively react with carboxylic acid side chains to form esters that can be utilized for protein labeling. The esterification reactions are easily reversible. Within the realm of top-down proteomics, the use of diazo-protecting groups could enable greater specificity and efficiency in the workflow. The typical proteomic workflow looks like this. There's an extraction of proteins from a cell culture, and those proteins are then isolated and purified. From there, they undergo a digestion in which the proteins are broken down into smaller fragments known as peptides. From there, the peptides can then be enriched and then separated through mass spec, where the data can be analyzed. In the optimization, the diazo compound would be used as a sort of unique molecular identifier that could lead to greater visibility of the amino acid residues in a sample via mass spec. This could enable greater accuracy when characterizing protein structure, and could also lead to a decrease in the ambiguity in regards to the localization of post-translational modification sites. Current work or future work of these chemistries could be the development of diagnostic tools for diseases that are specific towards post-translational modifications, such as NGLY1 deficiency and or other sort of genetic protein-based diseases.